अस्सलाम वालेकुम स्टूडेंट्स। होप यू ऑल आर फाइन। स्टूडेंट्स, आज हम बायो क्लास 11 चैप्टर नंबर 9 एक्सरसाइज लॉन्ग क्वेश्चन आंसर सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन इज लिस्ट द मैक्रो and micronutrients in plants, highlighting role of each. First of all, we see macronutrients. Nutrients which are required in large quantities are called macronutrients. These include nitrogen, and its role is in leaf growth, chlorophyll production, and protein synthesis. Next is phosphorus. It play a role in root development, flower and fruit formation, energy transfer. Next is potassium. Its role is in water balance, salt regulation, enzyme activation. Next is calcium. Its role is in cell wall development, root growth, nutrient uptake. Next is magnesium. Its role is in photosynthesis, cell wall structure, and enzyme activation. Next is sulfur. Its role is in protein synthesis, enzyme activation, and disease resistance. Next is micronutrients. These are nutrients which are required in small quantities. These include boron. It plays role in cell wall development, sugar metabolism, hormone regulation. Next is copper. Its role is in enzyme activation, connective tissue development, disease resistance. Next is iron. Its role is in chlorophyll production, photosynthesis, enzyme activation. Next is manganese, enzyme activation, photosynthesis, and disease resistance. Next is molybdenum. Its role is in nitrogen fixation, enzyme activation, and sulfur metabolism. Next is zinc. Its role is in enzyme activation, protein synthesis, plant growth and regulation. Next is chlorine. Its role is in water balance, photosynthesis, disease resistance. Next is nickel. Its role is in enzyme activation, nitrogen metabolism, plant growth regulation. These nutrients are essential for plant growth, development, and productivity. Deficiencies in any of these nutrients can lead to reduced growth, lower yield, and increased susceptibility to disease. Next question is explain the role of stomata and palisade tissue in exchange of gases in plants. Stomata and palisade tissue play crucial role in exchange of gases in plants. First of all, we see role of stomata. Gas exchange. Stomata, small pores on leaf surface, allow for gas exchange between plant and atmosphere. CO2 uptake. Stomata open to take in carbon dioxide for photosynthesis. Oxygen and water vapor release. Stomata release oxygen and water vapor as byproduct of photosynthesis. Next is regulation. Stomata regulate gas exchange by opening and closing in response to light, temperature, humidity, and carbon dioxide concentration. Next is role of palisade tissue. Location. Palisade tissue is a layer of tightly packed columnar cells just beneath the upper epidermis of leaf. Photosynthesis. Palisade cells contain many chloroplasts, making them the primary site of photosynthesis. Next is gas exchange. Palisade cells are in close proximity to stomata, allowing for efficient exchange of carbon dioxide and oxygen. Next is carbon dioxide diffusion. Carbon dioxide enters palisade cell through stomata and diffuses to chloroplast for photosynthesis. Next is interaction between stomata and palisade tissue. Carbon dioxide supply. Stomata provide carbon dioxide to palisade cell for photosynthesis, oxygen removal. Palisade cells produce oxygen as byproduct which is released through stomata. Water vapor transport. Water vapor produced during photosynthesis in palisade cell is transported to stomata for release. In summary, stomata regulate gas exchange while palisade tissue is responsible for photosynthesis. The close proximity of these two structures facilitate efficient exchange of gases, enabling plants to undergo photosynthesis and maintain water balance. Next question is relate transpiration with gas exchange in plants. Transpiration and gas exchange are closely related processes in plants. Transpiration. Water loss. Transpiration is the process by which plants lose water vapor through their leaf. Evaporation. Water evaporates from the surface of mesophyll cells, including palisade and spongy tissue into the multicellular spaces. Transport. Water vapor is transported to stomata and released into atmosphere. Gas exchange. Carbon dioxide uptake. Stomata open to take in carbon dioxide for photosynthesis, oxygen and water vapor release. Stomata release oxygen and water vapor as byproduct of photosynthesis. Next is relationship between transpiration and gas exchange. Shared pathway. Transpiration and gas exchange occur through the same pathway stomata. Water vapor release. Transpiration is primary mechanism for releasing water vapor into atmosphere, which is also a byproduct of gas exchange. Carbon dioxide uptake. Transpiration helps create a negative pressure which pulls carbon dioxide into leaf through stomata. Regulation. Stomata regulate both transpiration and gas exchange by opening and closing in response to environmental factors. Next is interdependence. Transpiration and gas exchange are interdependent processes. Changes in one process affect other. In summary, transpiration and gas exchange are intimately connected processes occur simultaneously in plant. While transpiration focuses on water loss, gas exchange encompasses the exchange of carbon dioxide, oxygen, and water vapor. The coordination of these processes is crucial for plant survival and productivity. Next question is describe structure of xylem vessel elements, sieve tube elements, companion cell, tracheids and relate their function and structure. Xylem vessel elements structure. They are dead, hollow tubes with thick secondary cell wall, lignified, waterproof and rigid, and have perforation plates for water flow. Lateral walls have pits for water transfer, and their function is to conduct water and minerals from root to leaf transpiration stream and provide structural support. Next is sieve tube elements. Their structure is they are long, narrow, living cells with a thin primary cell wall. End walls have sieve plates with pores for fluid transport, contain cytoplasmic strands and sieve element plastid. Function transport sugar, amino acid, and other organic compounds produced by photosynthesis, assimilates, play a key role in loading and unloading of sugars. Next is companion cells. Structure small specialized cells adjacent to sieve tube element have dense cytoplasm and numerous mitochondria connective to sieve tube elements via plasmodesmata. Function assist sieve tube elements in loading and unloading of sugars, provide energy and metabolic support. Next is tracheids. Their structure is they have dead lignified cells with thick secondary cell wall, have tapered end and pit for water transfer, often have helical or annular thickenings. Function conduct water and minerals in xylem similar to vessel elements and provide structure support. Next is in summary xylem vessel elements and tracheid form xylem, conduct water and mineral. Sieve tube element and companion cell form phloem, transport, assimilate, and structural features like lignification, plates, and sieve plates facilitate their function. Companion cells support sieve tube elements in slow transport. These specialized cells work together to facilitate transport of essential resources throughout plant. Next question is explain movement of water between plant cell and between cells and their environment in terms of water potential. Water movement between plant cells and their environment is driven by water potential, which is the sum of osmotic potential due to solute concentration, pressure potential due to turgor pressure, gravity potential due to height, metric potential due to interaction with cells. Water moves from an area of higher water potential to area of lower water potential between plant cells. Osmotic potential drives water into cell with higher solute concentration. Pressure potential drives water out of cell with high turgor pressure. Between cells and environment, water enters root through osmosis driven by lower osmotic potential in root cell. Water moves up the xylem due to osmotic potential gradient, transpiration pool, and coherent tension theory. Water leaves cells through transpiration driven by high water potential in cell, lower water potential in air. Key points Water potential is a measure of energy status of water in system. Water moves down its potential gradient from higher to lower potential. Plant cells regulate water potential to solute uptake, release, trigger pressure, and cell wall properties. This concept help understand water relation in plants, including water uptake, transport, and loss. Next question is: explain the movement of water in xylem through roots in terms of symplast, apoplast, and vascular pathways. Water movement in xylem through roots involves three pathways. First one is symplast pathway. Water enters root cell through symplast, living cells. Water moves through plasmodesmata, cell-to-cell connection, and cytoplasm. Symplastic movement is driven by osmotic potential and pressure potential. Next is apoplast pathway. Water enters the apoplast, non-living cell walls, and intercellular spaces. Water moves through cell wall, middle lamella, and intercellular spaces. Apoplastic movement is driven by osmotic potential and pressure potential. Next is vascular pathway. Water enters xylem vessel and trachees through pit and perforation plate. Water moves upward through xylem due to transpiration pool, coherent tension theory, osmotic potential gradient. Integration of pathways. Symplastic and apoplastic pathways coverage at xylem boundary. Water from both pathways enter xylem vessel and tracheid. Vascular pathway transport water upward to leave. Key points. Simplast and apoplast pathways are radial inward movement. Vascular pathways axial upward movement. Integration of pathways ensures efficient water transport from root to leaf. This explanation highlights the complex multi-pathway nature of water movement in xylem through roots. Next question is: explain the movement of water in xylem through tact mechanism. The tact, tension, adhesion, cohesion, transpiration mechanism explain water movement in xylem. First of all, we see tension. Transpiration create a negative pressure tension in xylem, pulling water upward. Adhesion. Water molecules adhere to xylem vessel walls and tracheids, allowing them to be pulled upward. Cohesion. Water molecules cohere to each other, forming a continuous column, allowing tension to be transmitted. Transpiration. Water evaporates from leaves, creating a continuous flow upward through xylem. Key aspects. Transpiration pull create tension during water movement. Adhesion and cohesion allow water to be pulled upward. Xylem vessel walls and tracheids provide a conduit for water movement. Continuous water columns enables efficient transport. Tact mechanism highlight the interplay between physical forces and xylem structure, enabling plants to transport water from root to leaf against gravity. Next question is: describe the mechanisms involved in opening and closing of stomata. Stomata opening and closing mechanism involve a complex interplay of guard cells. These are specialized cells which surround the stomatal pore. Turgor pressure, water pressure inside guard cell, ion transport, movement of ions like potassium ion, chloride ions across guard cell membrane, hormone signaling, abscisic acid and other hormones regulate stomatal movement, light and carbon dioxide sensing. Stomata respond to light intensity and carbon dioxide concentrations. Next is opening mechanism. Light stimulates guard cells to take up potassium ions. Increased trigger pressure causes guard cells to swell. Stomatal pore opens, allowing gas exchange. Closing mechanism. Abscisic acid signaling triggers potassium ion release from guard cells. Decreased trigger pressure causes guard cells to shrink. Stomatal pore closes, reducing gas exchange. Additional factors influencing stomatal movement include water availability, temperature, humidity, carbon dioxide concentration, circadian rhythm. Stomatal movement optimizes gas exchange, balancing carbon dioxide uptake for photosynthesis with water loss through transpiration. Next question is: explain the movement of sugar within plant. Sugar movement within plant occurs through two main pathways: phloem transport. Sugars produced by photosynthesis in leaves are loaded into phloem tissue. Phloem sap, a sugary liquid, flows through phloem vessels, sieve tubes. Sugars are transported to sink tissues, roots, stem, fruit for use or storage. Next is xylem transport. Sugars can also be transported upward through xylem tissue along with water and minerals. This occurs mainly in woody plants where xylem sap flows from root to leaves. Key mechanisms. Translocation. Sugars are moved from source tissue leaf to sink tissue like root, stem or fruit. Loading and unloading. Sugars are loaded into phloem tissue and unloaded at sink tissue. Pressure flow hypothesis. Phloem sap flows due to positive pressure generated by loading and negative pressure at sinks. Mass flow hypothesis. Sugars move through phloem due to concentration gradient. Regulation of sugar movement. Hormone signaling, auxins, cytokines, and other hormones regulate sugar transport. Sugar sensing. Plants sense sugar concentration, adjusting transport and metabolism accordingly. Sugar movement within plants is crucial for energy distribution. Sugars are allocated to tissues requiring energy, growth and development. Sugars support growth, differentiation and storage. Storage and retrieval. Sugars are stored in sick tissue and retrieved when needed. Next question is, explain movement of water into or out of cell in isotonic, hypotonic and hypertonic concentrations. First of all, isotonic condition. The concentration of fluids in the cell is equal to the concentration of fluid in surrounding solution. No net movement of water into or out of cell occur. The cell maintains its shape and size. Next is hypotonic condition. The concentration of fluid in cell is higher than concentration of fluid in surrounding solution. Water moves into cell through osmosis to dilute the fluid. The cells swell and increase in size. Next is hypertonic condition. The concentration of fluid in the cell is lower than concentration of fluid in surrounding solution. Water moves out of cell through osmosis to equalize the fluid concentration. The cell shrinks and decreases in size. In summary, isotonic. No net water movement, cell size remain constant. Hypotonic, water moves into cell swells. Hypertonic, water moves out of cell shrinks. This movement of water is crucial for maintaining cellular homeostasis, regulating cell shape and size, and ensuring proper cellular functions. Next question is, describe osmotic adjustment in hydrophytic marine and freshwater, xerophytic and mesophytic plants. Osmotic adjustment in plants refer to mechanisms used to maintain cellular water balance and trigger pressure response to changing environmental water availability. Here is a description of osmotic adjustment in different plant types. First of all, hydrophytic marine plants. They live in saline environment with high salt concentration, accumulate high concentration of ions, for example sodium and chlorine ions, to balance external osmotic pressure, synthesize compatible fluids, for example glycerol, proline, to protect cellular components, develop succulent structures to store water. Next is hydrophytic or freshwater plants. They live in freshwater environment with low salt concentration, maintain low ion concentration and high water content, use vacuole to store water and maintain turgor pressure, develop flexible cell walls to accommodate changing water level. Next is xerophytic plants. Live in arid environment with limited water availability, develop deep roots to access water deep in soil, store water in succulent stem leaf or root, produce thick cuticles, waxes or hairs to reduce water loss, use camp photosynthesis to conserve water. Next are mesophytic plants. Live in temperate environment with moderate water availability, maintain a balance between water uptake and loss, regulates tomatal aperture to control water loss, use osmotic adjustment, for example solute accumulation to maintain turgor pressure, develop moderate, succulent and water storage capabilities. In summary, hydrophytic plants, marine and freshwater, adapt to higher low salt concentration. Xerophytic plants conserve water in arid environment, and mesophytic plants balance water uptake and loss in temperate environment. These osmotic adjustments enable plant to thrive in diverse environment and maintain cellular homeostasis. Next question is, explain the osmotic adjustment in plants in saline soil. Plants growing in saline soil face high salt concentration, which can disrupt cellular processes. To cope, they make osmotic adjustments. First of all, ion exclusion. Plants limit salt uptake by excluding ions, for example, sodium and chlorine ions, from entering cell. Ion compartmentalization. Plants store ions in vacuole or organelles, reducing cytoplasmic ion concentration. Synthesis of compatible solute. Plants produce solutes, for example, glycerol, proline, betaines, that balance osmotic pressure without disrupting cellular processes. Accumulation of organic solutes. Plants accumulate organic solutes, for example sugars, amino acids, to contribute to osmotic balance. Salt glands or bladders. Some plants develop specialized structures to secrete excess salt. Deep roots. Plants develop deep roots to access less saline water deeper in the soil. Succulents. Plants store water in succulent structure, reducing water loss and diluting salt concentrations. Antioxidant production. Plants produce antioxidants to protect against oxidative stress caused by high salt level. Hormonal regulation. Plants regulate hormone production, for example, abscisic acid, ethylene, to coordinate osmotic adjustments. These osmotic adjustments enable plants to maintain turgor pressure, regulate ion balance, protect cellular processes, survive in saline environment. Keep in mind that different plant species exhibit varying degree of salt tolerance and osmotic adjustments may differ between species. Next question is, list the adaptations in plant to cope with low and high temperature. Low temperature adaptations. Cold hardening. Plants undergo physical changes to tolerate cold temperature. Antifreeze proteins. Some plants produce proteins which prevent ice crystal formation. Deep roots. Plants develop deep roots to access water and nutrients during frozen soil conditions. Dormancy. Plants enter a dormant state to conserve energy and protect sensitive tissues. Insulating structures. Plants develop insulating structures like hair waxes or scales to reduce heat loss. Soluble sugars and amino acids. Plants accumulate soluble sugars and amino acids to act as cryoprotectants. Winterization. Deciduous plants shed leaves to reduce water loss and protect birds. Next are high temperature adaptations. Deep roots. Plants develop deep root to access water deep in soil. Drought tolerance. Plants develop drought tolerant mechanisms like camp photosynthesis or succulents. Heat shock proteins. Plants produce heat shock proteins to protect against protein denaturation. Leaf orientation and movement. Plants adjust leaf orientation and movement to reduce heat absorption. Reflective surfaces. Plants develop reflective surfaces like waxes or hairs to reflect radiation. Transpirational cooling. Plants use transpiration to cool themselves through evapotranspiration. Next is thermal insulation. Plants develop thermal insulation like thick cuticle or bark to reduce heat transfer. Next are common adaptations. Stomatal regulation. Plants regulate stomatal aperture to control water loss and gas exchange. Water storage. Plants store water in succulent structures or leaves to maintain hydration. Osmotic adjustments. Plants make osmotic adjustments to maintain cellular water balance. These adaptations enable plants to survive and thrive in wide range of temperature. Next question is, explain the turgor pressure and explain its significance in providing support to herbaceous plants. Turgor pressure is pressure exerted by water within plant cells, particularly in the vacuole against the cell wall. It's caused by osmosis. Water enters the cell through cell membrane, increasing water potential. Cell wall rigidity. The cell wall resists the influx of water, creating pressure. Significance of turgor pressure in providing support to herbaceous plants. Cell shape maintenance. Turgor pressure helps maintain cell shape, preventing collapse. Stem rigidity. Turgor pressure contributes to stem stiffness, supporting herbaceous plants. Upright growth. Leaf expansion. Turgor pressure drives leaf expansion, allowing for photosynthesis and growth. Supports growth. Turgor pressure provides mechanical force for cell growth and expansion. Maintain plant posture. Turgor pressure helps maintain plant posture, resisting external forces like wind and gravity. facilitate water transport. Turgor pressure aids in water transport through xylem supporting plant hydration. Next is regulates tomatal movement. Turgor pressure influences tomatal opening and closure, controlling gas exchange. In summary, turgor pressure is essential for herbaceous plants, mechanical support, growth and function. It allows them to maintain their shape, grow upright, and respond to environmental factors. Next is describe the structure of supporting tissues in plants. Supporting tissues in plants provide structure support, protection, and facilitate water and nutrient transport. The main supporting tissues are, first of all, parenchyma, thickened cell walls with pectin and cellulose, flexible yet strong, found in stem, leaf, and petiole. Next are sclerenchyma. These are thickened, lignified cell wall, rigid, non-flexible, found in stem, branches, and vascular tissue, include fibers and scleroids. Next are xylem. Vascular tissue responsible for water and mineral transport, consists of trachea, vessel, xylem parenchyma, xylem fiber, found in stem, root, and leaf. Next is phloem. Vascular tissue responsible for sugar and nutrient transport consists of sieve cells, companion cells, phloem parenchyma, and phloem fibers found in stem, root, and leaf. These supporting tissues work together to provide mechanical support and structure, transport water, nutrients, and sugars, protect the plant from pathogens and environmental stresses, facilitate growth and development. In summary, the structure of supporting tissues in plants include collenchyma, sclerenchyma, xylem, and phloem, each with unique characteristics and functions that enable plants to grow, thrive, and respond to their environment. Next question is define growth and explain primary and secondary growth in plant. Growth in plant refer to permanent increase in size, weight, and volume of a plant cell tissue or organ resulting from cell division, expansion, and differentiation. Next is primary growth. Primary growth occurs at tip of root and shoot where meristematic cells, undifferentiated cells, divide and differentiate into new tissue. This type of growth is responsible for increase in plant height and root depth, formation of new leaves, stem and root, development of primary tissue, epidermis, cortex and endodermis. Primary growth is driven by apical meristem, root apex and shoot apex. Next is secondary growth. Secondary growth occurs in woody plants where new cells are added to existing tissue, increasing the girth or width of plant. This type of growth is responsible for increase in plant girth and diameter, formation of secondary tissues like xylem, phloem, bark, etc., development of wood and bark. Secondary growth is driven by lateral meristem, cambium, vascular cambium, and core cambium. Next is key differences between these two, primary growth and secondary growth. Primary growth occurs at tips, increase height and root depth, forms primary tissue, whereas secondary growth occurs laterally, increase girth, forms secondary tissue. In summary, primary growth drives upward and downward growth of plant, whereas secondary growth drives outward growth, increasing the plant's girth and diameter. Next question is describe role of apical meristem and lateral meristem in primary and secondary growth. Apical meristem and lateral meristem are undifferentiated cells that drive plant growth. First of all, we see apical meristem role in primary growth. Located at root and shoot tip, produce new cells through cell division, differentiate into primary tissue, epidermis, cortex, endodermis, responsible for increase in plant height and root depth, forms leaf, stem, and root. Next is lateral meristem. Role is secondary growth, located in cambium and core cambium, produce new cells that add to existing tissue, increase plant girth and diameter, form secondary tissue, xylem, phloem, and bark, develop wood and bark. Key differences between these two apical and lateral meristem. Apical meristem, primary growth, tip-based, increase height and root depth, whereas lateral stem, it is secondary growth, lateral-based, increase girth and diameter. In summary, apical meristem drive primary growth, forming new tissues, increasing plant height, whereas lateral meristem drives secondary growth, adding new cells to existing tissue, increasing plant girth. Next question is Explain how animal rings are formed in plants. Animal rings are formed in plants, specifically in trees and shrubs, through process of secondary growth. Here is a step-by-step explanation. Cambium layer. The cambium layer, a lateral meristem, is responsible for producing new cells that add to the existing tissue. Cell division. In spring, the cambium layer begins to divide, producing new cells that differentiate into xylem wood and phloem bark tissue. Xylem production. The cambium layer produces xylem cells which are larger and lighter in color, forming light-colored spring wood. Next is phloem production. As the growing season progresses, the cambium layer produces phloem cells which are smaller and darker in color, forming the darker summer wood. Growth slows. As the growing season ends, cell division slows and the cambium layer becomes dormant. Ring formation, combination of light-colored spring wood and dark-colored summer wood forms a visible ring marking 1 year of growth. Ring pattern, the pattern of light and dark rings repeats annually, forming a record of plant's growth history. Next is factors influencing annual ring formation. First of all, the factor which affect ring formation is climate, temperature, moisture, sunlight affect growth rate, water availability, drought or excessive water can impact ring width and density, nutrient availability, nutrient deficiencies can influence growth rate. Annual rings provide valuable information about plant age, growth rate, environmental condition, climate pattern. Dendrochronology, the study of tree rings, use this information to date events, reconstruct past climate, and analyze environmental changes. Next question is explain influence of apical meristem on the growth of lateral shoot. The apical meristem plays a significant role in regulating the growth of lateral shoots through, first of all, hormone regulation. Apical meristems produce auxin which inhibit lateral shoot growth, promoting apical dominance. Apical dominance. The apical meristem suppress lateral shoot growth, allowing the meristem to grow taller. Lateral meristem activation. When the apical meristem is removed or damaged, lateral meristems are activated, producing new growth. Lateral shoot release. As the apical meristem's influence weakens, lateral shoots are released from dormancy, allowing them to grow. Trophic correlations. The apical meristem influences nutrient distribution, affecting lateral shoot growth, morphogenetic fields. The apical meristem helps establish morphogenetic fields, guiding lateral shoot development. The apical meristem's influence on lateral shoot growth is inhibitory, suppressing lateral shoot growth, promoting apical dominance; regulatory, controlling hormone production, nutrient distribution, and morphogenetic fields; permissive, allowing lateral shoot growth when its influence weakens. Understanding the apical meristem role in regulating lateral shoot growth is crucial for plant breeding and pruning practices, horticulture techniques, and plant development and morphology studies. Next question is explain the role of important plant growth regulators. Plant growth regulators (PGRs) are chemicals which influence plant growth and development. Here is a brief overview of the role of important plant growth regulators. First of all, auxin, which promote cell elongation and cell division, regulate root growth, apical dominance, and tropisms, for example, phototropism, involved in fruit growth and senescence. Next is cytokinin. It promotes cell division and differentiation, regulates shoot growth, leaf senescence, and root development, involved in nutrient mobilization and stress responses. Next are gibberellins, promote stem elongation, leaf expansion, and seed germination, regulate flowering, fruit set, and senescence, involved in stress responses and nutrient uptake. Next is ethylene. It regulates fruit ripening, senescence, and abscission, involved in stress responses, wound healing, and disease resistance, promotes root growth and nodulation. Abscisic acid regulates tomaticoloyer, water loss, and drought tolerance, involved in seed dormancy, germination, and stress responses, promotes root growth and nutrient uptake. Next is Senosteroids regulate cell elongation, cell division, and differentiation, involved in stress responses, disease resistance, and nutrient uptake, promote seed germination and plant growth. These PGRs interact and coordinate to regulate various plant processes including growth, development, stress responses, nutrient uptake and allocation, reproductive processes, senescence and abscission. Understanding PGRs is crucial for plant breeding improvement, agriculture practices, horticulture techniques, plant biotechnology and genetic engineering. Next question is, explain type of movements in plants in response to light, gravity, touch and chemicals. Plants exhibit various type of movements in response to environmental stimuli including, first of all, we see light. Phototropism. Grow towards or away from light. For example, stem growing toward light. Positive. Photoperiodism. Response to day length, regulating flowering and dormancy. Next is gravity. Geotropism. Growth in response to gravity. For example, roots growing downward. Positive geotropism. And stems growing upward. Negative geotropism. Next is touch. Thigmotropism. Growth in response to touch or contact. For example, tendrils curling around sport. Thigmonesty, rapid movement in response to touch, for example, sensitive plant leaves folding. Next is chemicals. Chemotropism, growth in response to chemicals, for example, root growing toward nutrients. Hormone mediated movement, plant hormones like auxin, gibberellin, and cytokines regulate cell growth and movement. Other type of movement, nestic movement, non-directional movement in response to stimuli, for example, leaf folding in response to temperature changes. Circummutation, slow circular movement of stems and root, possibly due to internal rhythms. These movements help plants optimize light absorption and photosynthesis, maintain balance and orientation, respond to environmental changes and stressors, interact with their surroundings and organisms. Plants' ability to move and adapt is crucial for their survival and success in diverse environments. Next question is, classify plants on the basis of photoperiodism and give examples. Plants can be classified into three categories based on their response to photoperiodism. First of all, shade plants. Flowering is induced by short days and long nights, typically require less than 12 hours of daylight to flower, for example, chrysanthemum, soybean, rice, etc. Next are long day plants. Flowering is induced by long days and short nights, typically require more than 12 hours of daylight to flower. Examples include wheat, oats, barley, spinach. Next are day nutritional plants. Flowering is not affected by day length, can flower regardless of photoperiod. Examples include tomato, peppers, cucumber, and corn. Next question is, explain the mechanism of photoperiodism with reference to the mode of action of phytochrome. Photoperiodism is the response of plants to the duration of daylight which affect flowering, growth, and development. Phytochrome, a photoreceptor protein, plays a crucial role in this process. Phytochrome. Phytochrome exists in two interconvertible forms, PR red light absorbing form inactive and PFR far red light absorbing form which is active. Mechanism of photoperiodism. Light perception. Phytochrome absorbs light in the red 660 nanometer and far red 730 nanometer regions. Conversion. Red light converts PR to PFR while far red light converts PFR back to PR. PFR accumulation. During the day, PFR accumulates, promoting flowering in long day plants and inhibiting flowering in short day plants. Night break. A short period of darkness. Night breaks allows PFR to convert back to PR, inhibiting flowering in long day plants and promoting flowering in short day plants. Critical night length. The duration of darkness required to induce flowering in short day plants and inhibit flowering in long day plants. Mode of action. Phytochrome regulates gene expression influencing floral meristem identity. PFR promotes floral meristem formation in long day plants. Flowering time genes. PFR regulates expression of flowering time genes such as CO constants and FT flowering locus T. Hormone regulation. PFR influences hormone levels including gibberellins, auxins and cytokines. In summary, phytochromes act as a molecular switch converting between PR and PFR forms in response to light and darkness. This regulates gene expression, influencing flowering time, growth and development in plants. Next question is, define vernalization and explain the role of low temperature treatment on flower production. First of all, we see what is vernalization. Vernalization is the process by which some plants require a period of low temperature exposure to induce flowering. This process involves physiological changes that prepare the plant for flowering. Next is role of low temperature treatment. Low temperature treatment, typically between 4 to 10 degrees Celsius, plays a crucial role in vernalization by breaking dormancy. Low temperature breaks seed or bud dormancy, allowing for growth and development, regulating gene expression. Low temperatures alter gene expression, leading to the activation of flowering related genes. Hormone regulation. Low temperatures influence hormone levels, including gibberellins, auxins, and cytokinins, which promote flowering. Epigenetic changes. Low temperatures induce epigenetic changes such as chromatin modification, allowing for the expression of flowering related genes. Effects on flower production. Vernalization through low temperature treatment induce flowering, triggers flowering in plants that require vernalization, enhance flowering, increase flower production and quality in some plants, regulates flowering time, controls the timing of flowering, allowing for synchronized blooming, improve yield, increase seed production and yield in some crops. Examples of plants requiring vernalization include winter wheat, rye, barley, oats, some varieties of roses, carnations, and chrysanthemums. In summary, vernalization through low temperature treatment plays a critical role in regulating flower production by reducing. So स्टूडेंट्स हमारे ये लॉन्ग क्वेश्चन कंप्लीट हो चुके हैं होपफुली आपको समझ आ गए होंगे इफ यू हैव एनी डाउट यू कैन आस्क इन कमेंट सेक्शन प्लीज लाइक शेयर एंड सब्सक्राइब माय यूट्यूब चैनल थैंक यू